Okay, now this is the capsule stain. Capsules are a thick polysaccharide or sugar layer around the wall, outside the wall of the bacteria. And if a bacteria has a capsule, that means that it's more virulent. Virulent means that the degree to which a bacteria is pathogenic or can cause disease. So having a capsule allows those bacteria to resist phagocytosis. What is phagocytosis? If you remember that phagocytosis is when a cell engulfs another cell, and our white blood cells are phagocytic cells. They go around engulfing microbes and trying to get rid of them. Well, if a bacteria has a capsule and the white blood cell engulfs it, it resists that and it doesn't get broken down. Okay? Now that bacteria has just gotten past our immune system and can now cause disease. Okay? So the first thing in this procedure, we're on page 49, the second procedure, and it says to draw 2 circles, and I've already done that. Okay? That's only because we're using 2 different bacteria. Okay? And then it says add a loop full of Congo red. So I've already sterilized this, so I'm going to skip that sterilization step, but you would have sterilized it first. And so now I'm going to take this as Congo red, and I'm going to put a loop full of Congo red, and it says in each of those circles, but I'm just going to do one just to save supplies. Okay, now I have this dirty, so I need to sterilize it. Sterilize it because the Congo red is not sterile. Okay, so now that I've sterilized that, I'm going to get my bacteria, and remember I've got to let this cool off. So as it's cooling off, we're going to kind of tap the bottom of our tube of bacteria, and I really have bacteria in here today. And the 2 bacteria that we're going to use are S. salivarius and E. aerogenes. Okay. S. salivarius stands for Streptococcus salivarius, and you can imagine it's arranged in chains and it's circular, okay? And salivarius means it comes from your mouth, okay? So now we're going to get some of the bacteria. Okay, and then you're actually going to put the bacteria into the Congo red. I'm going to put it up here for now so that we can see what we're doing. Now you can see that the culture is red. Okay, now the next step says to let them air dry. Okay, so we would allow those to air dry. I'm going to go ahead and turn this off. After they have air dried, so this is still wet, but you would let it dry. And then it says fix smears with acid alcohol for 15 seconds. So did you see me heat fix that slide? No. This is one of the ones also, like the flagella stain, where you do not heat fix the slide. Okay, so now I'm going to fix it with, what did they say? Acid alcohol. And it says for 15 seconds. Okay, so remember this was already dry. Okay, so I wait the 15 seconds, and then it says, what color is your smear now? Well, it turned from red, now it's blue. Okay. So then we're going to let that sit for 15 seconds and then wash with distilled water. So we'll wash with distilled water. Okay. And then it says cover with acid fusion. Okay, remember earlier we used carbonyl fusion. Okay, now we're going to use acid fusion. Okay. This one actually doesn't in a dropper, so I'm just going to use a pipette for it. So then we're going to put the acid fusion on, and it says for a minute. Okay. Wash the smear with distilled water, so it sat for a minute. Wash with distilled water. Okay, and then it says examine under the microscope. Well, we'll go ahead and let it dry first. Okay. One of the cool things about this particular stain is that it's a negative stain, so it stains the background, and then the bacteria are not stained, and they look like clear halos They look like ghostly bacteria. The streptococcus, because it's in chains, actually looks like a string of pearls inside that dark background, and the background's usually a grayish or pinkish color. So it's a really, really cool stain to see.